Okay, the last thing that we need to talk about on this slide is why exactly do we not know exactly how many ATPs are produced at the end of cellular respiration from one molecule of glucose. There are a couple of reasons for this unknown or gray area in the accounting. If we were to take, let's see, a couple steps back, or actually, let's go forward. That would be better. There we go. All right. This slide summarizes the whole entire process of cellular respiration, plus it adds a couple of extra components that we haven't yet really looked at these similar figures that we've seen in previous slides, right? So we've got our mitochondria here, right? And then we have our stages of cellular respiration. Remember, glycolysis happens in the cytosol, produces a little bit of ATP, and it also grabs a hold of some of those high energy electrons for use later on here in oxidative phosphorylation. Glycolysis produces those two pyruvates. They go into the second stage of the cellular respiration, where the remainder of that leftover glucose molecule is totally degraded, and again we produce a little bit of ATP, those two ATPs at the end of the citric acid cycle. But most of the remaining energy from that original glucose molecule now is held in these electron carriers in the form of those electrons, which again will funnel into oxidative phosphorylation, and this is where we produce the remainder of the ATPs. And here's really where we get that about 26 to 28 ATPs. Why is that? Well, if we take a look over here back in glycolysis, NAD plus is reduced to NADH when it accepts those extra electrons from glucose. Now, NADH itself cannot cross the membrane of the mitochondria, right? You can't cross those two membranes, let's say, of the mitochondria by itself. So it needs to get those electrons to oxidative phosphorylation, which takes place in the mitochondria. How does it get those electrons in there? Well, there are going to be electron shuttles that help to pass the electrons from one electron carrier to the next across the membrane. So for example, the NADH here from glycolysis will come over and it'll drop off the electrons that it's carrying to those electron shuttles. And the electron shuttles will then pass them on to new electron carriers inside of the mitochondria that can then easily pass it on to the process of oxidative phosphorylation. That's great, no problem there, but you'll notice that while NADH does the drop-off here at the electron shuttles, what picks them up could be either NADH or it could be FADH2. We don't know. It's kind of the luck of the draw. Why does this make a difference? Now we have to go back a couple of slides. Let's go back to our electron transport chain. Let's go to bigger one. Bigger one? Bigger one. There we go. All right, so if we take a look again at the electron transport chain here, NADH and FADH2, they don't drop off their electrons at the same place. So it does actually matter whether those electrons get picked up by NADH or FADH2. So for example, NADH drops off its electrons at protein complex 1. So those electrons have a chance to help pump protons across using protein complex 1 and 3 and 4. They can pump a lot more of those protons across if they're dropped off at 1, right? Protein complex 1. On the other hand, FADH2, he kind of skips that very first protein complex. So the electrons that get dropped off by FADH2 here at protein complex 2, they go from 2 to Q to 3, so they pump protons here with 3, and then here again with 4, but they skip pumping any protons with protein complex 1, so they don't transmit as much of that energy across to that proton motive force, or that high hydrogen ion concentration. So it does matter, once those electrons get shuttled across the membrane, which of the two electron carriers pick up those electrons, because if NADH picks them up, they'll transmit more of their energy. If FADH2 picks them up, they won't transmit as much of their energy to that proton motive force. And we don't know which of the two electron carriers will pick up the electrons on the opposite side. So that makes it a little bit of a gray area in terms of the accounting of how many ATPs are produced per molecule of glucose. Also, when we were talking about the process of chemiosmosis, right, we have all these hydrogen ions that are stuck here in that intermembrane space. And I mentioned the only way that they can get across, or the only place they really want to go, is back into the mitochondrial matrix where their concentration is low. That's not exactly true. So remember, the mitochondria is within the cell, right? So if we were to imagine the outer membrane of the mitochondria out here, what's not to say that the protons or the hydrogen ions wouldn't want to move across the other membrane on the other side and move into the cytosol, where in the cytosol, granted, the hydrogen ion concentration is also low, just like in the mitochondrial matrix here. And in fact, some of these protons do actually, instead of moving down into the matrix through the ATP synthase, they actually escape the other way, going across the outer mitochondrial membrane. Now, this isn't necessarily lost energy, and I'll tell you why. Let's see if we can get back to this slide right here. So remember that pyruvate, right, that's formed at the end of glycolysis, it, in the presence of oxygen, will get shuttled into the mitochondria. This is a form of active transport. It's not just going to pass on its own. And the way that the cell pays for that active transport of pyruvate into the mitochondria is by coupling it with the outflow of those hydrogen ions from the intermembrane space out into the cytosol. So as those hydrogen ions flow out into the cytosol, they allow the bringing in of a pyruvate molecule. All right? So here's where, again, let's say here, some of those hydrogen ions, not all of them are used to make ATP through the ATP synthase. Some of them will go in the opposite direction and leave the mitochondria, go out to the cytosol to help bring more pyruvate in. Again, leading to our little gray area in terms of the accounting of how many ATPs can we actually produce using that one glucose molecule. Okay, this is also a great slide for you to review as you're going through that entire process of cellular respiration. It tells you exactly what's produced, the products. Maybe only thing missing on there might be some of the carbon dioxides that show, where the original carbons from the glucose that came in end up leaving. But other than that, the majority of the information is also there.